Recently, we've taken a look at how cells can use the information on their DNA in order to make proteins. Remember that this is basically a two-step process. During the first step, called transcription, the cell uses its RNA polymerase to make a messenger RNA copy of its DNA. Then in the second step, that messenger RNA travels to the cell's ribosome, where it undergoes translation to make the actual protein. If you consider a typical cell, it could have thousands of genes, and each one of those genes could potentially undergo this process of transcription and translation. Turns out, though, that not every single gene inside of a cell is turned on all the time. And basically the reason why that is because it saves energy. So turns out that not every single gene is needed all of the time. And so if a cell somehow had a way of turning off the genes that it doesn't need, that would save it quite a bit of energy. And so what we'll take a look at here is how cells can turn on and off their genes. Specifically, what we'll take a look at here in this video is how prokaryotic cells can turn on and off their genes. Later in class, we'll take a look at how eukaryotic cells can turn on and off their genes. The basic ideas are the same as far as how prokaryotic cells and eukaryotic cells turn on and off their genes, but there are some differences, and hopefully we'll tease out those differences when we take a look at this video and when we discuss it in class. So again, what we'll take a look at here is how prokaryotic cells can turn on and off their genes. The example of the prokaryotic cell that we'll look at is a type of bacteria called E. coli. You've probably heard of it. And the example of genes found in E. coli that can be turned on or off are the genes that encode for proteins that allow the bacterium to metabolize lactose. And so it turns out that several proteins are needed in order for E. coli to be able to break down and eat lactose. And so if there's lactose around, so in this picture what you're taking a look at here, this represents a single E. coli bacterial cell. If there is lactose in the bacterial cell's environment, here's what has to happen in order for this bacterium to be able to eat these lactose molecules. And so by the way, you might remember from maybe the second or third week of class that lactose is actually a disaccharide. So lactose is actually made up of one glucose molecule that's connected to a sugar called galactose. So glucose plus galactose, that whole thing is called a lactose molecule. There's a lot of energy that this bacterium can derive from this lactose molecule, and in order to get the energy out of it, again, a few proteins have to be made by this bacterial cell. One of the proteins that's required in order for this E. coli to eat that lactose is one that allows the E. coli to take the lactose in. So there has to be a pore protein available that again will allow that bacterial cell to take in the lactose. Not only that, but after the bacterial cell has taken in the lactose, in order to digest it, it has to be able to make a protein that serves as an enzyme that breaks apart that lactose so that glucose and galactose end up being produced. As soon as this glucose has been produced, that's what can enter glycolysis, and it's during glycolysis that this glucose can be broken down in order to produce ATP for a cell. Right, so again, E. coli, if there's lactose in its environment, it would be interested in bringing it in so that it can eat it and get ATP out of it. It turns out that the genes that are responsible for making these proteins, so this pore protein and this enzyme, those can be turned on and off. And so how about if we do this first? So let's name these proteins that are required in order for E. coli to break down lactose. This pore that allows the bacterium to take in the lactose molecules, it's called galactoside permease. Right, so that the name of this enzyme here, permease, that gives you a big clue as far as what it does. Right? So permease implies that this thing allows the bacterium to be permeable to lactose. Right? It allows E. coli to bring in lactose. The name of this enzyme that breaks down the lactose in order to form glucose and galactose is called beta-galactosidase. It turns out that there is another protein that's required in order for E. coli to be able to break down lactose. That third protein is called beta-galactoside transacetylase. And actually, people don't know exactly the function of this protein, but they know for sure that without this, the bacterium would not be able to break down lactose. Right, so in order for E. coli to break down lactose, these three proteins are required. What we're saying is that the genes that encode for these proteins can be turned on or off depending on whether the bacterial cell needs these proteins. Right? So, and when the bacterial cell needs these proteins is if there's lactose around. If the bacterial cell finds itself in an environment where there is no lactose, then it would be kind of a waste of energy, right, to make these proteins that break down the lactose. And so it turns out that E. coli can actually sense that. It can sense when there's no more lactose around, and in response to that, it'll shut down the genes that are responsible for making these proteins so that it can save energy. It's only if there's lactose in its environment that the E. coli cells end up making those proteins that will allow it to take in and then break down the lactose molecules. So E. coli, they're smarter than they look, right? So they can actually sense when there's lactose around and make the appropriate proteins that will allow it to break it down. If there's no more lactose, then it actually stops making those proteins. Pretty smart. So the bottom line here is that E. coli, an example of a prokaryotic cell, they can turn on and off their genes in response to the environment. What we'll take a look at next is how exactly do E. coli cells do this. If you remember genes, the molecule that contains the bacterial cell's genes, or any cell's genes, is the DNA. So to figure out how E. coli can turn on and off the genes that make these three proteins, how about if we take a close-up look at the DNA of E. coli, kind of like this. So we've now just enlarged that E. coli cell, and if you unwind that DNA that's found inside this cell, you'll find on there the genes that are responsible for making those lactose breaking down proteins. It turns out that the genes that make those proteins are all grouped together, and so we'll symbolize them. We'll use these light blue squares or rectangles to represent the genes that are responsible for making the lactose breaking down proteins. It turns out that these genes are grouped in such a way so that they share a single promoter and they share a single terminator region. In order for these genes to get expressed, in other words, in order for these genes to get transcribed and then translated, there are a couple of things that are needed. One of the things that's needed in order to transcribe these genes is something called Catabolic activating protein, or CAP for short. And it turns out that in order for this CAP to work, it has to be attached to a cyclic AMP molecule. The reason why CAP is required in order for these genes to be transcribed is because CAP helps RNA polymerase bind to the promoter. And so you remember that after RNA polymerase binds to the promoter region, what the RNA polymerase does is that it travels down the DNA until it hits the terminator region where it falls off. And what RNA polymerase does is that it reads the DNA and it uses that information in order to make messenger RNA. And again, remember, it's the messenger RNA that ends up going to the ribosome in order to make the proteins. And in this case, the proteins that are produced are the ones that we talked about previously. They're the ones that are needed in order for this bacterium to break down lactose. Right? So beta-galactosidase, galactoside permease, and beta-galactoside transacetylase are the three enzymes, or the three proteins that end up being produced by these genes. It turns out that the genes that are responsible for making each one of these proteins is given a name. So the gene that's responsible for making beta-galactosidase is called the LAC-Z gene. The gene that encodes for galactoside permease is called the LAC-Y gene. And the gene that encodes for beta-galactoside transacetylase is called the LAC-A gene. All right. So again, in order for these genes to get turned on, a couple of things are needed. RNA polymerase and this thing called the CAP. Before we sum that up, how about if we throw out a term here that's used to describe the way that these genes are arranged? That term is an operon. So whenever you're talking about an operon, that describes a group of genes that share the same promoter. And so again, that's what we see here. Because operons contain genes that share the same promoter and share the same terminator, that means that they can be turned on or off altogether. And so that kind of makes it easy to regulate these genes, to turn them on or off. A couple of other things about operons. Operons, they encode for proteins that work together. Right? So we're saying again that operons, an operon is comprised of a group of genes that are transcribed all together. And it makes sense to transcribe certain genes all together because the proteins that they end up producing work together. So in the case of this particular operon, the proteins that they produce are all proteins that work together in order to metabolize lactose. Right? So this protein, for example, would be useless without this one, and then vice versa. This one would be useless without this one. So it kind of makes sense to produce them as a group because they can only work when they're available as a group. One more thing about operons is that you find them only in prokaryotic DNA. So if you take a look at the genes on our DNA, for example, they're not all grouped together like this. Each one of our genes has its own promoter and its own terminator. Right? So again, this grouping of genes where a group can share a promoter and a terminator, that arrangement is only found in prokaryotic DNA. The take-home point here is this: operons, they can be turned on or off. When they're turned on, here's what they look like. It means that RNA polymerase is able to transcribe the genes in that operon. And we mentioned that there's something that's required in order to help RNA polymerase bind to the promoter. And that thing is this: it's that catabolic activating protein cyclic AMP complex. So that whole thing is required for this operon to get turned on, because again, it helps RNA polymerase bind to the promoter. So that's what operons look like when they're turned on. We're saying that this can get turned off though in certain situations, and so what we'll take a look at next is what the operon looks like when it's turned off. One of the major things that can turn off an operon is something called a repressor protein. And here's what repressor proteins do. Basically, they bind to DNA right here. So you can see the position of the repressor protein. It's positioned so that it's right between where the RNA polymerase binds and where the genes of the operon are located. And so if you have a repressor protein binding in this particular spot on the DNA, basically what it does is that it just blocks the RNA polymerase. It's not able to get to the genes that it's trying to transcribe. Right, so operons can be turned off. One of the major things that can turn it off are these things called repressor proteins. It turns out that, well, repressor proteins are a type of protein, and all proteins inside of a cell, the cell knows how to make those proteins as a result of having a gene that tells the cell how to make that protein. And the same thing is true here. So this E. coli cell is able to make this repressor protein because it has a gene that tells it how to make the repressor protein. The name of that gene is called LAC-I, and so LAC-I, that contains the information that tells the cell how to make the repressor protein. This spot on the DNA where the repressor protein sits, that's also given a name, and the name of that spot is the operator region of the DNA. Alright, so previously we showed you what has to happen in order for an operon to get turned on. Now we're showing you what happens when an operon is turned off. Basically, there's this thing called a repressor protein that just blocks the RNA polymerase from doing its job. So to sum that up, the lactose operon is turned off, again, when the repressor protein binds to a spot on the DNA called the operator. The gene that tells a cell how to make the repressor protein is called the LAC-I gene. And the thing about operons, so again, we're showing you what they look like when they're turned off, and then we showed you what it looks like when it's turned on. It turns out that in the case of this lactose operon, it turns out that its default position is off. So this thing is, this operon, the lactose operon, it's turned off all of the time. The only time when it would get turned on is if there's something that's in the environment of the E. coli that causes it to get turned on. And so you might have guessed that the molecule that causes the lactose operon to get turned on is lactose itself. And so we'll see why that happens right now. All right, so again, the default position of the lactose operon is that it's turned off. Right, so the cell is going to kind of save its energy, leave this thing off, unless it has a reason to turn it on. One of the reasons, or the main reason for turning on this lactose operon, is if the bacterium finds itself in an environment that has a lot of lactose molecules in it. Right? So if this bacterial cell sees that it has a lot of lactoses around, then it would be really interested in turning on this operon, because then it would end up producing the proteins that would let it eat all of those lactose molecules. So how does the bacterium then end up turning on this lactose operon whenever it sees that there's lactose in the environment? Turns out that these lactose molecules themselves cause the lactose operon to get turned on. And here's how. So if there's lactose around, the cell might have some limited ability to take some of them in. It turns out that once there's some lactose inside of the bacterial cell, there's a spot on the repressor protein that can actually bind to lactose molecules. And when that interaction happens between the lactose molecule and the repressor protein, it causes the repressor protein to change shape. And when it's in that altered shape, it can no longer bind to that operator region, so it ends up falling off. And so check out what just happened. So now that there is no more repressor protein blocking the way, that RNA polymerase is free to do its job, which is to transcribe all of those genes in the lactose operon. And once those genes are transcribed to produce messenger RNA, that can then go to the ribosome in order to produce those lactose metabolizing proteins. And so now that these proteins are produced, this bacterial cell now can really take in all of these lactose molecules, break them down, and get energy out of it. So to sum this up, we're saying that the lactose operon, it's turned on whenever there's lactose around to eat. Here's how that happens. It turns out that when there's lactose present, it binds to the repressor protein. When that happens, it causes the repressor protein to change shape. When its shape is changed, it ends up falling off the operator. And when it's no longer on the operator, that means that RNA polymerase can transcribe the lactose operon genes. Kind of the beautiful thing about the lactose operon is that it can get turned on, like we've just seen, but it can get turned off when it's no longer needed. And so this lactose operon, these proteins would no longer be needed if there's no more lactose around to eat. And so let's say that happens. Let's say that this cell ends up eating all of the lactose molecules. So now that there's no more lactose molecules around, it's kind of stupid to keep making these proteins if there's no more lactose for these proteins to break down. And so it turns out that when there's no more lactose around, it allows this repressor protein to kind of revert back to its original shape. When it's in its original functional shape, it's able to bind to that operator region again. After it binds to the operator region, that means that these genes can no longer be transcribed. So all of this stuff ceases to happen. So in other words, when lactose is gone, the cell is able to turn off this operon in order to save energy. So here's what we just said. When lactose is gone, that repressor protein once again can bind to the operator region because it reverts back to its original shape. And when that happens, it ends up turning off the lactose operon so that the cell saves energy. So what we've seen here again is that E. coli, it's not as dumb as it looks, right? It can respond to its environment and turn on or off genes as a result of it. And here's an example of a set of genes that E. coli can turn on or off in response to its environment. It turns out that when you're thinking about when you're considering the lactose operon, there's another factor that kind of plays a role in determining whether it's turned on or off. And here's that factor. It turns out that glucose has an effect on whether this lactose operon gets turned on or off. And so here's what we mean by that. If you have an E. coli cell that finds itself in an environment that contains both glucose molecules and lactose molecules, it turns out that the E. coli will end up shutting off the lactose operon. And so here's the kind of the logic behind that. If you compare glucose to lactose, turns out that glucose is a more optimal energy source, right? So if this bacterial cell was going to get energy from lactose, it would first have to break it down, right, so that glucose and galactose gets released. And only after that glucose is released can the bacterium use that glucose during glycolysis in order to make ATP. So if the cell is going to use lactose as an energy source, there's kind of an extra step that it has to do in order to get energy out of it, right? It has to first break down the lactose to release the glucose. Well, if the bacterium had access to glucose, then this would be the preferred source of energy, right? Because it doesn't have to do that first step of breaking something down. Glucose is glucose, so that can directly enter glycolysis so that the cell can get ATP energy out of it. So again, that's the rationale behind why if a bacterial cell has access to both glucose and lactose, it would basically ignore the lactose, concentrate on the glucose because it's a better source of energy. So then how does a bacterial cell, if it has access to both glucose and lactose, how and why does it end up turning off the lactose operon? And here's how that happens. We've already seen what happens if there's lactose around, right? It can enter the cell, bind to the repressor protein, which causes it to change shape. When it's in that altered shape, it slips off of the operator region, so that RNA polymerase looks like it can transcribe these genes. So if you're taking a look at the operon right now, you see that it's in the on position, right? RNA polymerase can transcribe those genes. There's nothing blocking the way. But what we're saying, though, is that if this bacterial cell is in the presence of glucose, this lactose operon ends up getting shut down so that the bacterium can concentrate on making glucose metabolizing proteins. So here's how it ends up getting shut down. So if there's glucose in the environment, that can end up entering the cell. Once that glucose is inside of the cell, remember that can enter glycolysis, and during glycolysis, ATP is produced. If you remember back when we were talking about cellular respiration, one of the things that's needed in order to make ATP is AMP. Right? So basically when ATP is produced, two phosphates are added onto adenosine monophosphate in order to make it adenosine triphosphate. So if there's glucose inside of the cell, again that enters glycolysis. During glycolysis, ATP is produced. But in order to make ATP, this AMP is required, right? So if there's glucose inside of the cell, this AMP ends up getting used up because it's being used to make ATP. It turns out that if there's no AMP around that's available to bind to this cap protein, that cap protein ends up changing shape. And it turns out that in this altered shape, it can no longer bind to the DNA molecule, so it ends up slipping off. Remember that the job of that cap protein was that it helped the RNA polymerase bind to the promoter. So if cap is no longer bound to the DNA, that makes it a lot harder for RNA polymerase to bind to the promoter. So chances are that it'll also fall off. And if there is no RNA polymerase that's available, or if RNA polymerase can't bind to the promoter, that means that these genes found in the lac operon never get turned on. And so again, we're saying that the presence of glucose ends up turning off the lactose operon, and here's how the cell ends up doing that. All right, to sum up, the presence of glucose causes levels of cyclic AMP to decrease inside of the cell. When there's no cyclic AMP, that means that catabolic activating protein can no longer maintain its shape, so it ends up falling off of the DNA. Once cap is no longer on the DNA, that means that RNA polymerase has a harder time binding to the promoter. If RNA polymerase can't bind to the promoter, that means that that means that the lactose operon ends up shutting down, so that the bacteria in this case can concentrate on making glucose metabolizing proteins instead of lactose metabolizing proteins. And again, the rationale behind all of this is that glucose is a more optimal energy source than lactose. So if the cell again has access to both types of sugars, it would save its energy by not making lactose metabolizing proteins, and instead it would concentrate on making glucose metabolizing proteins. So again, here's kind of one major way that prokaryotes can turn on and off their genes